Building water systems refer to the water distribution systems inside buildings. In technical terms, they are also known as premise plumbing. This system includes pipes, storage tanks, taps, showers, and cooling towers. They are important because they supply water for many daily use, including drinking, washing, cleaning, and also cooling. Although the water entering the building has already been treated, the plumbing system itself is not completely sterile. Microorganisms can still be present and enter the system through the water, pipe materials, or the surrounding environment. Once inside the system, certain conditions can support their growth. For example, warm temperature, low water flow, and water stagnation can make it easier for microbes to survive and multiply. This is especially common in areas of the system where water stays still for a long time or where the disinfectant level becomes lower. The main concern is that these microorganisms can attach to the inner surface of the pipes and form biofilms. Biofilms are important because they are usually hidden inside the plumbing system, so they are not easy to see or remove. From this, there are three main problems that can happen. The first problem is related to water quality, where biofilms can change the taste, odor, and clarity of the water. For example, when water flow suddenly becomes strong, the biofilm can break off from the pipe surface. This can cause water discoloration and may also release metals such as zinc and manganese into the water. So even if the water looks clean at first, disturbance in the pipe system can cause visible change in water quality. The second problem is related to pipe efficiency. Biofilm can grow better on rough and corroded pipe surface. Over time, corrosion and biofilm buildup can make the inner pipe surface rougher and narrower. This can reduce water flow and make the plumbing system work less efficiently. If the buildup becomes severe, it may also affect the long-term condition of the pipe. The third problem is related to human health, where biofilm can act as a hiding place or reservoir for opportunistic premise plumbing pathogens, OPPP. For example, including Legionella species, Pseudomonas species, and Mycobacterium species. These microorganisms can be harmful, especially for people with weaker immune system, elderly people, and hospital patients. People may be exposed through direct contact with contaminated water or by inhaling tiny water droplets from showers and taps. This is why biofilms in building water system is not only an engineering issue, but also a public health concern. So in summary, building water systems can support microbial growth because they provide suitable conditions such as warm temperature, stagnation, and pipe surface for attachment. Once biofilm form, they can affect water quality, reduce pipe efficiency, and also increase health risk.
I'll discuss biofilms in building water systems, including why microorganisms form them, how they develop, and the role of extracellular polymeric substances or EPS. So, why do microbes build microbial cities? Imagine this. Even after water pipes are cleaned and disinfected, microorganisms can still survive and grow on the inner pipe surface. These microorganisms do not live alone. Instead, they gather and build protected communities called biofilms. A biofilm is a structured community of microorganisms attached to a surface and embedded within a self-produced EPS matrix. In building water systems, biofilms commonly develop inside pipes, storage tanks, cooling towers, and faucets. Microorganisms form biofilms because this structure helps them survive unfavorable conditions. It protects them from disinfectants and environmental stress, improves nutrient capture, and allows communication between cells. However, biofilms can reduce water quality, promote pipe fouling and corrosion, and create potential public health risks. Therefore, understanding biofilm formation is important for maintaining safe and efficient water systems. Now let us look at how a biofilm develops. Biofilm formation occurs through five main stages. The first stage is initial attachment. Free-swimming microorganisms approach a surface and attach quickly through forces such as van der Waals forces, electrostatic attraction, and hydrophobic interactions. At this point, the attachment is still reversible. Next is irreversible attachment. The cells produce adhesins and surface proteins, allowing them to attach more strongly and begin colonizing the surface. The third stage is EPS production. The microorganisms secrete extracellular polymeric substances, which form a matrix around the cells. This is followed by maturation, when the biofilm develops into a complex three-dimensional structure. Water channels also forms within it, allowing nutrients to reach the microorganisms. Finally, during dispersion, some cells detach from the mature biofilm and spread to the other surface, where new biofilms may begin to develop. The survival of a biofilm largely depends on its EPS matrix. EPS is a protective material produced by microorganisms and consists mainly of polysaccharides, proteins, lipids, and extracellular DNA. EPS performs several important functions. First, it provides protection by limiting the penetration of disinfectants and shielding microorganisms from environmental stress. Second, it traps and retains nutrients, ensuring that the cells have access to essential resources. Third, it provides structural support by holding the microbial cells together and helping the biofilm remain attached to the surface. Finally, EPS retains water and prevents the microorganisms from becoming dehydrated.
Moving on to the common physiological traits of biofilm forming microorganisms and how different microorganisms specialize within biofilms through their redox and metabolism. Biofilms are communities of microorganisms that attach to surfaces and become embedded within a self-produced protective matrix. Although different microorganisms may live in a biofilm, they share several important physiological traits that help them survive. The first trait is surface attachment. Microorganisms use structures such as villi, fibrae, and adhesin to attach to surfaces and initiate colonization. This is the first step in biofilm formation. Once attached, they begin producing extracellular polymeric substance or EPS. EPS acts like a protective glue that holds the microbial community together. It provides structural stability, improves surface retention, and creates a diffusion barrier that limits the penetration of harmful substances. The third trait is enhanced stress resistance. The EPS matrix protects microorganisms from disinfectant, antibiotics, nutrient limitation, and other environmental stress. This is one reason why biofilms are often difficult to remove from water systems and medical devices. Another important characteristic is cell-to-cell communication through quorum sensing. Microorganisms release signaling molecules that allow them to communicate and coordinate gene expression. This helps regulate biofilm maturation and community behavior. As biofilm develop, oxygen penetration becomes limited in a deeper layer. This creates oxygen gradients within the biofilm. As a result, cells in the inner regions often switch to slower metabolic activity, which increase their resistance to environmental stress and antimicrobial treatments. There are different microorganisms specialized in biofilm according to their physiological capabilities and metabolic strategies. The first microorganism is Pseudomonas aeruginosa. It is considered one of the strongest biofilm formers and produces large amount of EPS. It can survive in low oxygen or hypoxic regions of biofilm by switching its metabolism. Under aerobic condition, it uses glucose or other organic compound as electron donor and oxygen as the terminal electron acceptor. However, when oxygen becomes limited, it can use nitrate as an alternative electron acceptor through nitrate respiration. This metabolic flexibility allows Pseudomonas to continue generating ATP and survive in oxygen-depleted environments. Next is Legionella pneumophilia. This microorganism is commonly associated with warm water systems such as cooling towers, water tanks, and shower systems. Unlike Pseudomonas, Legionella mainly relies on aerobic respiration using amino acids and organic compounds as electron donors and oxygen as the electron acceptor. It is particularly adapted to environmental persistence and can even survive within protozoa, which provide additional protection in aquatic biofilms. The third microorganism is Escherichia coli, which is often used as an indicator of fecal contamination in water system. E. coli can form mixed species biofilm and adapt to changing oxygen conditions. Similar to Pseudomonas, it uses oxygen during aerobic respiration but can switch to nitrate reduction when oxygen becomes scarce. This flexibility helps E. coli survive in deeper biofilm layers and other oxygen-limited environments. Overall, redox reduction are essential because they generate ATP, which provides energy for microbial growth and maintenance. The oxygen gradient inside biofilm create different metabolic niche, allowing microorganisms to use alternative electron acceptors such as nitrate when oxygen is unavailable. This adaptation contributes significantly to biofilm persistence, stress resistance, and long-term survival. Biofilm forming microorganisms share common physiological traits such as surface attachment, EPS production, stress resistance, and quorum sensing. However, each microorganism has developed specialized redox strategies that enable it to thrive in different biofilm environments, making biofilm highly resilient and difficult to eliminate.
Biofilms can have several negative impacts on infrastructure. First, they cause infrastructure problems by promoting corrosion and clogging pipes. Second, they lead to water quality problems because biofilms can release contaminants and unpleasant odors into the water. Finally, they pose health risks since harmful microorganisms can grow within biofilms and potentially spread waterborne disease. Although biofilms can be harmful, they also provide important benefits. In wastewater treatment, biofilms help break down organic pollutants efficiently. In water purification, biofilm is intentionally cultivated and helps in remove contaminants and improve water quality. Biofilms are also used in environmental remediation, where microorganisms degrade toxic chemicals and help restore polluted environments. To manage biofilms effectively, an integrated control strategy is needed. Chemical methods include chlorination, ozone treatment, and hydrogen peroxide treatment to kill microorganisms. Physical methods such as pipe flushing, mechanical cleaning, and high-velocity water flow help remove biofilm buildup. Engineering controls focus on using corrosion-resistant materials, reducing stagnant water zones, and improving system design. Lastly, environmental controls include temperature management, regular water quality monitoring, and maintaining adequate disinfectant levels to prevent biofilm growth.